Hey everybody, this is your last lecture for the biochemistry unit. Yay! And it is all about enzymes and chemical reactions. Remember, enzymes are a type of protein and we are working with them in lab over and over again. So chemical reactions are an interaction between two chemicals. Chemical bonds are either broken or formed, and the two parts of the chemical reaction are the reactants and the products. The reactants work together to form an end product. Some chemical reactions release energy, and these are called digesting polymers, and some chemical reactions require an input of energy, and these are called building polymers. All chemical reactions have what is known as an activation energy. This is the energy needed for a chemical reaction to start. Most reactions in our bodies require high energy to occur quickly enough to keep a cell working properly. If you start off with the same reactants here, the activation energy is on the red line is very, very high. That's the top of the bump. When you add an enzyme, that is the result in green, it lowers the energy of activation and allows you to accomplish changing the reactants to the product with much less energy being used. So enzymes are catalysts. They are proteins that do the body's work, and they help to speed up chemical reactions by interacting with substrates to lessen the energy needed. Most enzymes have names that end in ACE, A-S-E. Remember, we learned sugars have names that end in OSE, O-S-E, and enzymes have names that end in ACE. Remember that ACEs break down OSEs. This is a picture of an enzyme. The one that is lighter blue is the enzyme, and the active site is the area where the enzyme can fit in, the substrate. The substrate, or the specific substance that the enzyme is going to work with, is what attaches and binds to the active site and then can change. Okay, the enzyme in this picture is the big like purplish globule, and all enzymes have a very specific shape. Only specific substrates will fit into them. They will fit into the active site, and the enzymes and substrate fit like a lock and key, and that's called an induced fit. So a lock has a specific key that will work in it, and not just any key will do. In the same way, an enzyme has a specific substrate that it works with, and not just any substance will fit. Activation energy, here's another, we already looked at one type of graph to show this. This is another one. Activation energy is the energy needed to start a chemical reaction. An energy bump you must jump over to get going. An example would be pushing a rock downhill. The enzyme acts as a crowbar. So imagine this, if you were at the top of the hill and you had to push a really huge boulder down the hill. Now, it had a little bit ways to go before you could get it down the hill, and this thing weighs, it's almost as big as you, so you are just trying really, really hard to get it rolling, and it's taking you a lot of energy, and you're straining and having to push as hard as you can, and maybe you even need to recruit some help. Well, the protein will act like putting a crowbar underneath it. Now, if you've got the crowbar underneath it and you can step on that, you can get that big boulder rolling down the hill with using much less energy. Here is a picture of a little alien pushing a rover, a space rover, up over this hill. So this is all of the energy required to push the rover up the hill on the top, and the potential energy released by the downhill run, but it's not captured by useful work. So that's without an enzyme. The alien pushing, working really hard without the enzyme. And then the enzyme is the little man in black here that made this a whole lot easier by just making the hill a whole lot shorter. So now it doesn't take hardly as much energy for that alien to get the rover up over the hill. So make sure you take a moment and draw this graph. And write down what effect does an enzyme have. This is the same graph we saw before. The red line is your changing your reactant to your product, but it has this really high amount of energy needed for it to do the activation energy. The green line right here shows the result of using an enzyme and lowering that activation energy. The initial beginning reactant and the end products are both the same, with or without an enzyme. 4 features of enzymes. Enzymes make reactions happen faster. They're called catalysts. They do not alter or use up enzyme molecules. Reactions do not alter or use them up. So you remember, if you're watching, the enzymes in the proteins change the hydrogen peroxide, we could use that same liver substance over and over again for the hydrogen peroxide reaction. We were not using up the enzyme, we were just changing the peroxide. The same enzyme usually works for both the forward and reverse reactions, and each enzyme has a specific shape and recognizes and binds to a specific substrate. Factors that influence some enzyme activity: temperature, pH, and salt concentration. If your enzymes or your proteins aren't at a specific temperature, pH, or salt concentration, then they will denature. If they are too acidic or too basic for their living environment, or if you have cooked the meat, then you're going to do something called denature the proteins, and that is going to unwind them. So think of that tertiary protein structure that's all coiled and beautifully folded like a ribbon, and just unwinding it and pulling it apart, and then the enzymes cannot do their job. The effect of temperature on pH, this graph shows this. Small increases in temperature increase molecular collision or increase the rate of reaction, but high temperature will disrupt the bond and destroy the shape of the active site. So it's showing as you increase small amounts, it's good, it helps the reaction, but once you get too hot, there's a level here where once it's too hot, and it just destroys all the proteins and they can't work at all. pH. Which curve likely represents an enzyme in the stomach? Why? So think about this for a little bit. You should write down your answer. This is a pH scale from 1 to, about 11 here. The pH scale goes from 0 to 14. And this is the level of activity. The higher the level of activity, the better. So the one that represents enzyme in the stomach is going to be this one over on the left that's kind of a lavender color. It has the highest activity in the very acidic portions of the stomach. That's because the stomach acid, gastric acid, is very, very low on the pH scale. It is extremely acidic. Which curve represents an enzyme in the blood? Why? I hope you were able to guess. It is this middle one. Blood pH is 7.4. So right here at 7, with the highest activity, that is where the enzymes are going to be. What you can think of is on your own is maybe where an enzyme, that functions in a basic environment, what type of enzyme might that be?